This talk is gonna be about reverse engineering and bacteria. Two things you don't usually associate with each other, but I mean, how do bacteria store their immune system? Or how do microbes store their immune system, and where? And how do bacteria bootstrap themselves? And how do we find out? Well, we can sequence the DNA, the code of life, the code that is closest to our hearts, literally, and we can reverse engineer it. And how this happens, Bert Hubert is gonna tell us today. So, a bit of applause, please. Because he's a regular at this event. He has already held 2 great talks as SHA, 1 great talk as MCH, and another great talk today. And I encourage you to check them out as well. You can get them on Media CCCDE, for example. And they're all great talks about DNA, molecular biology, and I guess life in general. And now for reverse engineering a bacterium. Thank you.
Well, thank you so much for showing up for a talk about molecular biology and reverse engineering. And the wonderful thing is, of course, there is about no thing is as much fun as reverse engineering. And I'm going to offer you the chance to reverse engineer life, and I hope that you will take that up. The credits, many of the pictures you see come from Wikipedia Commons. Sometimes I can credit them in the page, sometimes it doesn't fit. By all means, visit this QR code, because there you get the PDF of the presentation that for most slides has additional URLs or links or extra explanations. The good news is, even if you don't feel like scanning this QR code, it will repeat at the end of the presentation. So you can sort of wait it out and see it if it's worth your while. As noted, you can ask, for the online audience, you can ask questions, and I hope you previously scanned the QR code already, because this is the pretty brief time to scan the QR code. But if you are online, please do also know that you can ask questions. So, who am I? I could talk a lot about who I am, but the most important thing is, am I full of shit or not? And I didn't study biology. I got into that later in life. Am I a scientist? I don't know. But I have these 2 presentations, these 2 articles in Nature. And I think maybe I'm a scientist, maybe I'm not a scientist, but I at least managed to fool a lot of people. So I think it's legit what I'm telling you. And nothing I am telling you is particularly new or controversial. It's just rarely explained this way. Because I said this yesterday also, but I'm just going to repeat it. The technology of life is so incredibly awesome. We have found our own ways of making USB sticks, which can store like terabytes of data. And then we find that nature can store gigabytes of data using 40 atoms per bit. That's like really nice. That is extremely tight. And they can also build storage that works for 200,000 years. And I'm pretty sure that if I leave my SSD drive around for 2 years, it will be empty. But the DNA goes on after 200,000 years. So the technology of life is extremely interesting, and I hope to tell you a little bit about it. I'm going to skip this one. What I'm going to talk about. You know the joy. You have a new machine, it has firmware, and you wonder what is inside this machine. Does it run Linux? What is it firmware? Is it encrypted? Can I modify the firmware? And DNA is our firmware. And we're going to analyze it to see what can we learn. Meanwhile, I'm just going to tell you lots and lots of cool DNA stuff, because I cannot stop myself. So I must tell that to you, and I hope you enjoy it. And I also have an open invitation. The biology world, the DNA world needs you. Because there's no reverse engineer like you. And there is stuff that we don't know what it does, and maybe you can help. So to briefly recap, DNA, the letters A, C, G, and T, we organize them in chromosomes, because if they get too big, we split it. Nature splits it up. Apparently, nature too has issues organizing data. But the weirdest thing about this is that all of life, every mushroom, every bacterium, every virus every plant uses the exact same DNA. And it's also compatible with each other. So a virus can inject DNA into us and it will work. And that's not what we want, by the way. But sadly, it does work. And we could put our DNA in bacteria, and then the bacteria go run our DNA and make insulin for us, which is nice, actually. So thank you, bacteria. But the interesting thing is this has been going on for like 4 billion years. And I cannot read the DVD disc from 10 years ago. I no longer have the equipment. Apparently, life figured out how to store data and continue doing that for like 4 billion years. Important about this slide is we live in a so-called 3-domain system of life. And you might think that the system is plants, animals, and I don't know, maybe us. But that's not how it works. The 3-domain system of life is 2 different kinds of bacteria, the archaea and the real bacteria, which look a lot like each other and are totally different for reasons no one understands. And then in the middle is the fusion of these 2, and that's us. But the interesting thing about this, these archaea and bacteria are very simple. All of them are very simple. And all of us, dus the plants and the cucumbers and stuff, are exceedingly complex. And there's nothing in between. We'll get to that, but I think it's important. Is DNA digital? I think it is. We're going to skip this slide. This is what it looks like. DNA, when you have a really good microscope, you can look at it, and it's a piece of tape. It's just a linear thing. Sometimes it's a circle, and that's where the information lives. And the interesting thing about this, it is storage. It really is storage, and we'll show you how much data is on there. But it is not addressable storage. We cannot ask what is on DNA position 5023, because there are no numbers on there. So it's like a piece of tape with data, but no sectors, no markers, so you never know where you are in DNA. But they've learned to live with that. So this is a very simple virus, hepatitis B, and you can catch it if you go on holiday. And it's a very small virus, and it is 800 bytes large. And these are the 800 bytes. And you can just read it, and it's safe to read. You can just read, it doesn't jump through your eyeballs into your brains. But this is really all there is to it. To the point that if I go to a DNA printer, and I hear someone here is building a DNA printer, and we need to talk, you can upload this file to it. Out comes a little bit of DNA. You put this in a human cell, not your own cell, don't do that. And it will generate hepatitis B viruses for you. You can really start the hepatitis B virus from absolute scratch, from a floppy. But is life really digital? Because maybe it works for a virus. So at one point, people gathered a lot of money. And I mean a lot of money. And they took a bacterium on the left. Oh, it's not here. And they took out the DNA of the bacterium. So they had an empty bacterium. Then they designed a whole new genome based on other bacteria. So here's one I made earlier. I forked it, I changed it a bit. And they inserted that DNA into the empty bacterium. And then nothing happened. Because in theory, everyone is sort of sure that if you create DNA from your computer and you put it in an empty bacterium, it should work. And when they did this, after they'd spent like $30 million, it didn't work. And whenever you try to bootstrap something, you learn. Because truly knowing stuff means bootstrapping it. And it turns out that empty bacterium that they used had an unknown antiviral mechanism. And when we put in the new DNA, that mechanism said chop, 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 not doing it. Okay? There was a learning, a teachable moment. So then they removed that antivirus system as well, put the whole new firmware into the empty bacterium, and it bricked. It did nothing. And then they had to go back, and they found out that in the whole genome that they had inserted, they had missed one letter. And you might think, well, you can do without one letter, how bad can that be? But because there is no addressing on DNA, if you remove one letter, all the other instructions move up a little bit. And then nothing worked anymore. Well, finally, they fixed that too, put the fixed DNA in the bacterium, and it came to life. Does that mean that we can create life completely from scratch now? It does not yet mean that, because you have to start with the bacterium. And then you have to empty it out, put new firmware in there. I'm sure that one day we will skip that step as well, but this is as digital as we can get it right now. So, let's take a look at the genome files. And you can download these. It's not easy to do, I'll show you how, but you can now download 50,000 bacteria and their complete DNA. And 50,000 is enough that you can do real statistics on that. Now, the first thing you can, of course, do is if you have a bunch of firmware files, you can say, how big are they? Because the bigger, you learn something. And then we find that almost all bacteria are around 1 megabyte in size. Some of them are 2, some of them are a little bit smaller, but there are no super big bacteria. The biggest one is like almost 3.5 megabytes. Apparently, that is the limit for how much firmware you can put in a bacterium. And you cannot put more in that. Interesting here on the left, you see this arrow, and you see there is a minimum genome size for a bacterium. There is no bacterium smaller than that, from which we can derive that there is a core set of genes that if you do not have that, you are not life. And that's very interesting, but it does not get smaller than that either. So that's the minimum viable bacterium. Most of these are parasites who live inside us and who have outsourced most of the work to us. So they can do with very little firmware. Why do bacteria care? Because that will be important later on. Copying DNA costs time. A typical bacterium takes 20 minutes to copy itself. And which means that after an hour, we have had 3 doublings. So if you start with 1 bacterium, after an hour, you have 8. And after 2 hours, you have 64 or whatever, and eventually you have billions. The faster a bacterium can replicate itself, the more copies it will make. And in this little graph, I sort of shown you that if you have a bacterium that doubles every 20 minutes, it will grow like this. And at this point, it vastly outnumbers the bacterium that copies every 40 minutes. So for bacteria feel the need for speed. They will remove any bit of DNA that is not useful, because that allows them to copy faster. So we're going to put the smallest complicated life form. This is yeast. And yeast is actually in our family. It's not a bacterium, it's actually complex life. And you can see it already has internal departments. It has a nucleus, it has a vacuole, it has a mitochondrion, doesn't matter what it all means. But this is like a company with several departments. And the bacterium is like really just the single company person, of the single person company. Everything is just in there. You can see the DNA, that the black squiggles, that's the DNA. And where does it store the DNA? Well, yeah, over here and over there. It just floats there. So here you can see 2 ways of making life. The simple way and the somewhat complex way. And there is, in gene size, in terms, there's nothing in between. And you can sort of compare this with companies, that if you are a small company, you can be very effective. If you are a large company, you could be very effective, but mostly are not. But there is a sort of interim place where it's just like difficult, because you're not yet a really large company, you're still a small company, and life is a bit inconvenient there. And we see that here as well. So we have some further life forms. This is a meat-eating plant. So don't worry, it's not going to eat you. It's a very small plant. But this actually has a 14 megabyte genome. So you can build a whole plant out of 14 megabytes. Well, it's a very simple plant, but it already has an outrageous number of departments in there. It's like this has structure. You need to fill out forms to get anything done in this cell. And but this is nothing. This is nothing. The next least complex animal is the pufferfish. And it's beloved of OpenBSD fans, and it has the tightest animal genome of the whole Earth, the whole world. So this is already good. For 100 megabytes, you get a whole fish. And it's very compact. It's worth it to get 100 megabytes for a fish. And from this point on, it becomes ridiculous. So I draw a human being here, and this human being has 750 megabytes of DNA. Now, of course, this human being feels a bit more interesting and complex than a pufferfish, but it's a lot of DNA. I mean, compared to the plant that could do it with 14 megabytes, it's a lot. But this is like nothing. We have this fish, and this is now a logarithmic graph, where you can see where we go from the simple bacteria, then we have the yeast, we have the meat-eating plant, we have the lovely fish, we have a human being, and then we have this fish. It does 33 gigabytes of DNA. And we really don't know why. And it is a nice fish, I have to admit that, but I'm not sure if it's... I mean, this is almost as big as a game update. Yeah. So anyhow, this is the distribution of genome sizes, and already we've learned something. You can be very simple life, or you can be somewhat complex life, or you can go completely overboard. But there's a little overlap. It's not worth it to be the most complex bacterium. It's not worth it to be the simplest complex life. So genomes, you can go to this site from the US government, and it used to be great, and we're already worrying that it will go away. Because that's how they roll in the US these days. But you can log in here on their FTP server, and they also have an rsync server, and there you can download all 55,000 genomes that we have, mostly of bacteria. And I also made a site which gets you the same, lots of interesting data, but in a format that you will all be more familiar with, CSV files. So all the graphs you see come from the CSV files that you can download from skewdb.org. And that graph, if we have time, we get to that graph, and otherwise you'll have to wait to tomorrow, because tomorrow there is an overflow after party at 3 p.m. for if you have more questions. So let's dive into the 50,000 binaries we just downloaded, but sadly these binaries are all ASCII. And they look like this. This is the actual DNA of a simple bacterium. And this is what these files look like. And you see that they start with ATG, and I can disclose to you that's actually the start of gene marker. So it's sort of fun that they actually start there. It's not always the case. It's all ASCII. It's just letters. And there are 50,000 of these files. You can look at them. It's called sequence gazing. And you look at it, you're like, can I see some patterns in there? And I can guarantee you, you cannot. After a while, if you stare enough at this, you see a license plate and you start reading it as DNA. But I think this is also well known to reverse engineers. They also look at hex streams and start seeing things. The other trick that we often do, when I get a file and I don't know what it is, I try to compress it. Because if it compresses, there is apparently structure in there. If it does not compress, it's likely encrypted. DNA does not compress. And this is very frustrating for everyone, because people were like, I see all these repeating patterns. I think it should compress. It does not compress. So these are the 50,000 gzip files. Because there are only 4 letters in there, you should get like 75% compression just based on Huffman, and that's also more or less what you get. But otherwise, bacteria do not repeat themselves. So, why do we store it as ASCII? Because it's a ridiculous waste to use a whole 8 bits of ASCII to store 2 bits of DNA. And it turns out there is a reason for that. Sometimes the DNA machine is not sure which letters there are. And that's the N in between. This is where the DNA machine says, I'm sure there's DNA there, but I don't know what it is. And we cannot fit the N in the 2 bits. So all our DNA files we have on disk are 4 times too large. It's a good thing that disks are cheap these days. So this is where it gets a little bit complicated. DNA is in a way rather weird. I mean, I said it's like a tape, but it's a tape where you can store data on the top side of the tape or on the bottom side of the tape. And, but we also have rules that if there is an A on the one side of the tape, there has to be a T on the other side. And if there's a G on one side, there has to be a C on the other side. This is a redundancy mechanism. Because if any of these letters somehow becomes damaged, life can repair that letter by looking at the other side, which is neat. It's nice. But where it gets a bit weird is that you can read this DNA. It actually works in two directions. So you can read it from left to right, and that's where it starts with ATGC. But on the other hand, you can read it from the other end. So it's point symmetrical, as indicated by that little star. Now, the biology people are sometimes ridiculous, but they're luckily not that ridiculous. So the file on disk you get is only one part of the DNA. Because if you have the top part, you can imagine what the bottom part would look like. And, but on the top is what the actual DNA file looks like, and the bottom is the same DNA, exactly the same DNA, but written in the other direction. This turns out to be important for following slides. Well, nature, like I said, this is DNA, and there are some genes on one half of the DNA, the top half. There are also some genes on the bottom half, which actually then go the other way. And nature is such a lead hacker that it sometimes makes DNA where you can have two genes that share the same letters except shifted a bit. And that's a very lead hack. Only viruses really do this because they want to make their DNA smaller. Now it's going to get a bit interesting. We look at a bacterium and we say how many letters A are in there, how many letters C are in there, how many letters T, how many G. And then you find that many bacteria have 25% A, 25% C, 25% T, 25% G. But there are also bacteria which consist for almost 40% on the letter A. And also 40% on the letter T. And interesting, if you spy with your little reverse engineering eye, you might notice that some of these graphs are actually almost identical. The A graph is identical to the T graph. And the C graph and the G graph also are identical. So what we have learned from this is that bacteria apparently have the same amount of A as the amount of T, and the same amount of C as G. And if you plot it like this is from almost 50,000 genomes. This is the fraction that is C, that is the fraction that is G. And you see that these fractions are almost identical. And this is called the second Chargaff parity rule. And here you can probably, many of you cannot see it, but here you can see that the deviation, the difference between the C fraction and the G fraction is typically between minus 0.5% or plus 0.5%. And the interesting thing is we do not know why this happens. So this is where you study the firmware and you see, well, if there are this many instructions of this, apparently there are also this many instructions for Y, and it's always almost the same number, and no one knows why. And I'm leaving this little mystery with you, because we hope to solve it one day. But it gets a bit weirder. You can also take longer strings of DNA, and then imagine what they would look like on the other side of the DNA. That's called reverse complement. And we saw that there's the same amount of C as G, which is weird, but it turns out that even if you do this for longer strings, there is the same. If you scan over DNA and you take all combinations of 3 letters, you find that there are almost exactly the same amount of combination CTA and TAG. And again, this holds for all the different combinations of 3 letters. They're all the same. And again, we do not know why. If you ask a biologist why this is, you will often get the reaction, oh, but that's completely logical. And then you can ask a bit more, say, yeah, but how did it happen? Then they say, oh, it's so trivial, I cannot even explain this right now. And you can look up the Wikipedia page, it is full of this stuff that's, yeah, it's completely logical and completely unexplained. enjoy. This is what it looks like. You see the number of CTA and TAG is the same, and you see all these numbers, all these bars are almost exactly the same, which is actually sort of more interesting than exactly the same. Now, this holds true for all forms of life, even for longer strings. So this is all 6 long DNA strings, and yeah, everything is on this straight line again. And why 2025? So they organized this whole festival so that I could ask this question. I'll skip this a bit. And okay, now we're going to look at one bacterium. We take the DNA and see which 6 letter combinations. So I first take the first 6 letters, and then I shift 1 position, and I take that 6 letters, and I check how often do these strings occur. And then we find that the string CCT-AGG in a whole bacterium happens 32 times. It's actually 16 times, because I just realized there's a bug in my software. It's nice that I remember that during the presentation. But it's 16. So the sequence CCT-AGG barely happens. Meanwhile, we have the sequence CGC-CAG, which happens 10,000 times. So apparently this is a very popular sequence. And again, we can make a graph of this. And here you see this CCT-AGG sequence, and you cannot even see it on the graph. It only happens 16 times. And then on the far right of the graph, you have CGC-CAG, and it happens 10,000 times. Now, I've told you a lot of stuff that we do not understand, but this is the one that we actually do understand. What we see from here is that there are forbidden sequences, that this bacterium says, I'm like the knights who say "ni," who cannot stand the word "it." This bacterium says, I cannot stand the word CCT-AGG. Why would it do that? And it turns out it is a very devious idea. So, this guy is for real. This is a bacterial virus. You might think this is some sort of schematic, an artist impression. No, this is what the damn thing looks like. And we know that because we have the DNA source code for this virus. And so we know exactly which proteins these are and how they work. This guy really looks like this. And I'm very happy that he infects bacteria and not me. And also that he's like 100 nanometers large. Here on the right, for those of you lucky enough to be able to see on the right, these are some actual electron micrographs of what this thing looks like. And what he does is this. He takes this body, he sits on a bacterium, and he injects his DNA in there. And because DNA is completely compatible across life, the bacterium will say, okay, I have my instructions, I will get going. Sadly, the instructions are destroy yourself and turn yourself into a virus factory. And it does that really well. So once this DNA is in there, it will start making more of these guys, and until the bacterium is full, and then it explodes. And then a billion new viruses come out. This is a bad thing for a bacterium, and it does not want that. So what does it do? It actually has little enzymes in there, denoted here by these Pac-Man icons, that know that there are specific DNA sequences which are the enemy. And this little Pac-Man guy knows that he should destroy CCT-AGG. Once every 4,096 DNA combinations, that combination will happen by accident. That means that viral DNA is restricted. It will only work as long as it avoids the sequence CCT-AGG and a whole bunch of other sequences. Well, it's nice that this way the bacterium can defend itself against the virus, but it must make very sure that it does not destroy its own DNA. Because these little Pac-Man guys are not very intelligent. So they will gladly rip up your own bacterial genome just as happily as they would rip up the viral genome. So what we observe is that these bacteria have these sequences which they themselves avoid a lot and also protect with a sort of protective paint. And then if a virus wants to successfully infect that bacterium, it somehow has to know which sequences to avoid and which, some luck, it will never find out. And we have found this out because you can look this up. We found this out using this simple table. It says, are there any sequences that never happen? We found a bunch of sequences that never happen. And we also know they do not occur in many viruses. And this is the way it works. Now, these DNA cutting things are very useful for genetic manipulation. So when this was discovered, everyone was like, oh, thank you. Now we can manipulate DNA. And remember the sequence that we put in there, CCT-AGG, and this is where you can buy it. New England Biolabs, and there it says CCT-AGG. You can buy this, you get an invisible amount of DNA, and you can use that to cut DNA wherever you want. And we discovered that just by counting letters, which is rather nice. So, what does the DNA actually build? What we know is that DNA gets converted through complicated means into amino acids. And there are 20 amino acids that we know. And the DNA actually instructs the cell, combine the following amino acids together, and then you will get a new virus or maybe eyes or other stuff. This is the 1D printer that actually prints life. Now the question is, of course, which DNA letters correspond to these amino acids? And there is this glorious table. And I'm so in awe of this thing. This is like a very technical table. It says, for example, if you have the 3 DNA letters CCC, you must be a pro. So I'm not sure if there's anyone from CCC here, but nature agrees with you. This is a 4 billion year old table. Have you ever seen a 4 billion year old table? I mean, nothing lasts that long, but we know this is actually how it works. There are some extremely minor variations in this table, but they are like so small that you're like, yeah, that actually only confirms how universal this table is. And there are also start codons in there. For example, ATG, this little arrow means the gene starts here. This is like a super Rosetta Stone, except then like billions years old. So, on the top left, we have DNA from this bacterium that we've been looking at, and you can see it starts with ATG, which is this start codon. It's nice. We then want to know not only what are the DNA letters, but also what proteins does it make. But because this is the interesting part where the biology happens. And this is a conversion from the DNA, and then you have to know where does the gene begin, where does the gene end. This top file format is called FASTA. But it's not very fast, so I don't know why they call it that. But and this is the GFF format, and really, these biologists, they should have a very stern talking to, because they keep inventing the most terrible file formats. So this one is partially space delimited, partially tab delimited, and also semicolon delimited. And parsing this thing is sure hell, but the worst thing they did is they actually made addressing for DNA so that you can refer to a position in the DNA, and it is 1-based. That's just terrible. I mean, it's the original sin. But yeah, apparently they start counting from 1 in biology, we start counting from 0, and this has actually led to mountains of bugs in bioinformatics software. So, there is a piece of software that does this analysis for you. And what it does, it looks at DNA, and there are some magic strings in there. For example, TTG, ACA, and TATAT, sort of happy announcement of a new gene. And these letters have to be there because then the cell mechanism knows, okay, a new gene is beginning. Okay? And at the end, then there's a Shine-Dalgarno sequence, and I also found it sounded very exotic to me. It's like Shine-Dalgarno, and it's 2 Australian blokes. So, and they also found a sequence, AGG, which is when this 3D printer gets the hots for. And at the end of a gene, it's very interesting, this is where life is a bit more analog than usual. You know that DNA, that an A attaches to a T and a C to a G. If you make a palindromic DNS sequence, then it will stick to itself. And this is actually at the end of bacterial genes. And why is that there? To make sure that that machine falls off. Because you don't want all these machines sitting on your DNA. So this is the off-ramp, where they say, okay, now get out. So this is how you can recognize a gene, by those magic sequences and by this loop at the end. And this is by now a well-solved problem. Almost. And then you get this. This is a visual display of one bacterium where I took the 3 million bytes of DNA and I represented them as this spiral. It is not actually a spiral, but it allows you to see all genes. Every stretch you see here, a blue stretch, this is one gene or perhaps a few genes that are very close together. The blue ones are on one side of the DNA and the orange ones are on the other side of DNA. And you see that they cluster together a little bit. And so this is a nice thing to see. We also see some bootstrapping mechanisms. That thing up there is the 3D printer that actually prints life. It's nice. It can print copies of itself easily, unlike the shitty human 3D printers. The problem is, of course, still, if you have a new cell, how do you build the first one? And it turns out there's a bootstrapping mechanism by which they can build a proto 3D printer using these red bits. So it's nice that we can see that. Also interesting to see already is there are parts of the spiral which have no genes on them. But that's weird, because we learned that bacteria want to have the smallest genomes possible. They do not want to have any useless stuff in there. We should all admire bacteria, because they're like a billion-year-old software project and there is no bloat. Yeah, we can't have that. How do you convert these genes, these gene letters, into the gene begins here, the gene ends there? There's a tool for that called Prodigal. And it's so old that it ships with Debian. And also I want to congratulate everyone with Debian on their Trixie release today. It is a glorious day. I love Debian. So I'm sorry for making fun of it. But this is a super standard tool. You can install that today. Take a bacterium, analyze it, and it will say, I found the following genes for you. And it looks like this. It takes a single minute. You run it. Prodigal, in, out, genes. It thinks for 1 minute, and it says, okay, I found 3,500 genes for you. This is the absolute standard tool in this field. It's getting a bit old, but it's like the sort of the SendMail of things. It's sort of always it's the VI. It's the VI of gene analysis. But I was a bit worried about the tool because it's written in artisanal C. And it's quite terrible C. So I thought, let's run this extremely important program which sort of underlies all bacterial genomes. I'm going to run it in Valgrind. And it said, oh, we have a conditional jump or move depending on uninitialized values. It's not the sort of news that we want, actually. And so I looked into it, and there was a very obvious bug in there. And I opened a pull request for them. And I filed it. It's already 4 days ago, and they didn't apply it yet. I'm not priorities for these people. But okay, if you get hurt by viewing memory unsafe code, you should not watch now, because this is what they did. If the genome was a bit larger than they thought, they would reallocate new space and not zero it. This is a classic mistake, of course, but it is rather scary that this happens in such an important tool. And this is actually the second time that I found this in such an important tool. There's one other tool called SAMTools, which is used in the medical field a lot, and it cannot distinguish between a malformed line and end of file. So if it analyzes your medical data and it sees one malformed line, it will just say, oh, apparently that was the end of the file. And you best hope that your disease was not beyond that. It took them 9 years to close my pull request on that. So if you had any faith in this world, sorry. Well, this is the most weird and fun bit, I think. These bacteria have been waging war with viruses for the past 2 billion years. And they've become rather good at it. Well, you know, when I showed that spiral, there were some of these bits in the DNA that are not genes and we don't know what they are. And for like 20 years, people had been looking at sequences where they saw a sort of fixed bit of DNA and then what they called a spacer. And then again, the same bit of DNA would repeat, and then there would be all kinds of random DNA, and then it would repeat, more random DNA. And because these people are not computer professionals, they thought, well, this bit in the middle, they called it a spacer. They said, we don't know what it is, it's probably not interesting. Because it's random. They said it's random, so it's probably not interesting. And they spent 20 years looking at this stuff. And then at one point, someone who was wise looked at this spacer and they said, I recognize this as viral DNA. So in the bacterium, there is a list of pieces of viral DNA. And it turns out this is an immune system. If a virus gets into a bacterium and it is lucky, then it will catch the DNA of that virus and recognize it as the enemy and store a copy of that DNA in there so that in the future, when that same virus infects it again, it can destroy it. So this is a virus signature file. And it contains copies of maybe 12, sometimes 20 viruses. This is interesting. Bacteria have an antivirus memory of 20 viruses. Now, and this really blew me away. There's a paper from 2002, and that says, well, we find that these spacers, you can find them between this element, CGG, TTA, TCC. And I thought, let's open up the DNA file for my bacterium and search for that, and boom. The non-inverted things are actual viral DNA that this bacterium protects itself against. So in the bacterium, we can find a memory of the viruses that it had encountered over the past, we don't know how long years, actually. This is a very forensically interesting thing. It's like when your server is there, and you look at it, and it contains a file that, yeah, my previous generations have been hacked by this malware. It's super interesting. And it carries this around, and we can just see it. And this is how it's stored. But this is so obvious that you can see it with the incredibly powerful forensic tool called LESS. And I'm pretty sure there is more of this out there. One interesting question, of course, is, if this DNA says you must destroy the DNA that says AG, TTT, CCG, TA, why does the bacterium not immediately destroy itself? It's the same thing if you pass around signature files with malware signatures, they might get flagged as malware themselves, which is a known problem. And it turns out that the bacteria are rather clever. So what they have done, they said, yeah, actually, we always pick the signature where we do not include all the DNA, but we will only match if, in addition to our signature, you then get the sequence AGG. So there is an implicit part of the signature that is not in the bacterium, but says, I know about that anyhow, and this is how it makes sure that it will destroy the virus, but not its own signature file, which is neat. Let us not do this. Well, a little bit then. I told you that bacteria have the same amount of C and G, and we don't know why. But it gets weirder. At the beginning of the chromosome, there is more C than G. And after halfway, there is more G than C. And again, if you ask biologists, why is this the case? They go like, yeah, either it's super simple or we don't know. And to this day, this is what my paper in Nature Scientific Data was about. So I told these biologists, I said, look, you don't know why this is going on. And I made a database for you with 50,000 ways in which you do not know what is going on. And interestingly, that's now like 3 years ago, and there have been 9 follow-up papers by other scientists, and they have also said, yeah, that's weird. And I really hope that we can figure this out. I have some theories, and oh, these are some very nice graphs, which you will have to look up. Actually, I found these graphs on the Wikipedia, and I thought they're nice, and then it turned out I had made them. But I didn't know anymore, so I too should have a good memory, like bacteria. Blah, 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 skip that one. I'm going to repeat this one. We saw that in one of these white spaces, after 20 years of not getting it, people suddenly found an antiviral system that was like super advanced, because it's so advanced that we took this CRISPR antiviral mechanism and we're now using that to cure genetic diseases. Because again, the bacterial DNA also works in human beings, and we can make a piece of DNA that says, if you find the following wrong bit of DNA, you must cut it to pieces. And we can do this in human beings now, or better, we can use it with blood that we got out of human beings, because it's scary. But accidentally, in one of these white spaces, we found one of the biggest revolutions in medicine ever. There are more white spaces. And this is, I think, where the reverse engineering community, the hacker community, once you gain access to these bits of white space, you could start saying, well, are there patterns in there? Or can I, with my computing knowledge, take these bits of white spaces and compare them to all the other genomes we know? Maybe there are clues there. And I'm sure that there are like 7 Nobel Prizes in this graph. If only we had the digital skills to do this analysis coupled with sufficient biology knowledge. Because that is a bit of a problem. We have like real biologists, and they are experts in their field. They know everything about genes and molecules and stuff. They're wonderful. They know fuck all about computers. And that's a bit of a problem, because they have interesting intuitions. They might think, I would love to investigate this, but it sounds like it's too complicated. Meanwhile, we have all these computer people, bioinformaticians, that know surprisingly little about biology. So they could do all kinds of interesting things if only they had the intuition. If only they had seen cool talks where people said, look, these are the cool biology things going on, instead of having to go to biology school and learn the 5 different kinds of muscle fiber, which is like stupid boring compared to this stuff. So I think that if we could change this distribution with biologists that know a little bit about computers and computer people that know a little bit about biology, if we could make this overlap far more, then many great things could happen. I'm not sure why this is funny, but you will probably let me know afterwards. But I make a very serious point here, because it turns out it is far easier to have computer people and teach them the coolness of biology than to take biologists and turn them into computer programmers. Because we have done that, and that leads to these scary GitHub pull requests. I need to temper this a little bit. The people over here are actual geniuses. Do not get me wrong, because they are fighting to understand all this stuff. They invent incredibly smart experiments to do this stuff. So these people are great. They are the real heroes. But I can only imagine how much faster things would go if they knew what was possible over here, and if the reverse engineers, the great reverse engineers of this world, would get their hands on DNA. I think with that, we have reached the end. These are some open questions. Why does the G happen more at the beginning of the genome than at the end? Can we figure that out? Why do we have the same amount of C's as G's? What is in that white space? And the biggest question, why is so much of the software crap? I hope you can help with that. And with that, I thank you so much for your attention.
Applause.
One final thing. Thank you so much. I skipped a bunch of slides. Tomorrow, 3 p.m., Cassiopeia tent. We're just gonna nerd out. So show up, ask a million questions. And I'll also do the missing slides, and I hope that for some of you hardcore nerds, that will be fun.
Awesome. Thank you, that's a great talk. I think we have 2 more minutes for questions, so if you do have a question, please queue up at the microphone or write it in the REC. Don't be shy.
Yeah.
All right, one question, please.
Yeah, hi. First of all, great talk. It's always a joy to hear you talking. You mentioned the biology people are not aware of what computer people can do. Have you held a presentation like this on a biology conference?
I actually have. I took my research that I published and I went to them, and I found that they were like completely surprised that a computer person showed up at their conference. So yeah, I have tried to work with it. There have been initiatives to try to make it simpler for them, but it's not a question of a better UI. It's a question of really understanding it. So, but we are trying.
Okay, thank you very much.
Next question, please.
Thank you for a really interesting talk. I hope I'm not the only one here who knew you as the guy who twitters about Dutch government doing stuff with people's data. How do you have time for this?
Well, let me give you and 500 of my best friends apparently the honest answer. I do not actually have a job. And that means that the time that you sit at the office or working for a boss, I sit writing new blog posts. And that gives me a lot of time. So if you did not have a job, you would also be able to do this, I think.
Okay, maybe 1 or 2 more questions. Let's see.
I was just going to ask, for the histograms of the sets of 3 or 6, was that just splitting it into 3 without any frame errors?
Exactly.
Yeah,
it's just a sliding window.
So have you done analysis on if it was offset, or is that just...
But you can do separate analysis with framing, and then you get completely new mysteries. Okay. So please show up tomorrow. We can geek out on it.
Thank you.
Okay, thank you. Sorry, we're out of time. Thank you again.
I will be outdoors over there, so we can talk on this. Thank you so much, people. Thank you.